This is TWIS. This Week in Science, episode number 735, recorded on Wednesday, August 21st, 2019. What's in a brain? Hey everyone, I'm Dr. Kiki, and tonight we will fill your head with brains, bones, and koala poo. But first,
TWIS is supported by listeners like you. We thank you for your support. We really couldn't do it without you.
Disclaimer. The following program is not intended for all audiences. If you are curious-minded or like learning about curious things, if you like thinking logically or speculating wildly about where logic may lead, if you like methodical inquiry or asking questions with no established pathways to find the answers, if you like science, you have come to the right place. If, on the other hand, the very idea that the world is more complicated than you might have assumed fills you with dread and you enjoy a good scare, you have come to the right place. If, on the other hand, you are physically allergic to knowledge, wonder, and unbiased curiosity, by all means, stay tuned and document your symptoms so that we may find a cure for you. Come to think about it, this show actually might be intended for all audiences. So welcome everyone to another episode of This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. This week in science. What's happening? What's happening? What's happening? This week in science.
Good science to you, Kiki and Blair.
and a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about all the science that we found fit to bring to the show this week. You know, there are hundreds, thousands of papers published weekly around the world. So, you know, we don't bring quite that many, but I like to think that the stories we bring are always interesting. So what did we bring tonight? I brought stories about brains. That's pretty much it. Brains and behavior. There we go. That's all I have. Justin, what did you bring?
I've got a Toxoplasmystery solved. I've got Summering by Lake of Bones, Underwater Archaeology, and Mongolia.
You've got Mongolia.
Mongolia.
It's like Trump wanting Greenland. I don't know. Okay.
Yeah, but I made my deal.
And Blair, what is in the animal corner?
Oh, I brought some koala poop. I brought locust face plants. And I brought some bioluminescence.
Ooh, that's always luminescent and bio at the same time. It's very cool.
It's always luminescent bioing, yes. All right, let's jump into all of the science goodness. So we want to jump into things, but before we do, I want to remind you that it would be wonderful if you would check out TWIS.org. That's where you can find out how to subscribe if you are interested over on YouTube or on the Google Play podcast portal. And what's the other one? Oh, yeah, the Apple podcast directory. The three big ones. We are also available on Spreaker, Stitcher, TuneIn, Radio.com, Pandora. You name it, we're pretty much there. Just look for This Week in Science, and we hope that you will find us. TWIS.org, that's our website. Jumping into the science, I am going to start with, brains. Like I said, lots and lots of brains. So first we're going to start in the distant past with the question of how did we evolve from our ancestors? This giant cranium. Look at the size of that cranium. Well, that giant cranium that humans happen to have, it didn't come out of nowhere. The presiding... Huh?
No, never mind. I was going to say where it actually comes out of, but it doesn't. It's not what you meant.
Yeah. This head of ours, this brain of ours that allows us to speak and cognate, have all sorts of wonderful behaviors and abilities. It came down a long line of evolution, and that is the presiding view, that it was a line. And we kind of think of the picture of like, hey, look at our ancestors with their really tiny brains. And then look, the brains got bigger with time, and then suddenly a massive leap in processing power and abilities of behavior, and our brains are huge. Well, researchers just published this week a new study that is in Science Advances, led by researchers from the American Museum of Natural History, the Chinese Academy of Sciences, and the University of California, Santa Barbara. And they posit, dun dun dun, that the brain got big and small and big and small and didn't grow in this linear way. Surprise, surprise to our TWIS listeners, right? If you've been listening to Justin for so many years, listening to the stories that we bring, it's a braided stream, right? And it's not going to be a linear progression. Right. Why did we think it would be? Well, it's the limitation of the fossils that we've had. And so now they say that over the course of anthropoid history, that the path of that brain evolution is a lot more complex and interesting than others had previously recognized. The researchers say human beings have exceptionally enlarged brains, but we know very little about how far back this key trait started to develop. This is in part because of the I can't speak tonight, the scarcity of well-preserved fossil skulls of much more ancient relatives. So the older they are, the more broken up into lots of pieces they are. They're not well preserved. And so it's been harder to know exactly how big things are, how they've grown. We don't have that many. So it's a very limited sample set. Well, these researchers were looking at a 20 million year old fossil that was discovered in the mountains of Chile. It's this little tiny skull known as Chilicebus carasoensis. And over 30 years, these researchers have been looking at lots of different fossils, and they say that Chilicebus is one of those rare and truly spectacular fossils, revealing new insights and surprising conclusions every time new analytical methods are applied to studying it.
But this isn't Chile at 20 million years ago. How is this at all relevant to the brain of human peoples?
Because it's an anthropoid. It's an ancient ancestor of ours. Huh. And so previous work gave a rough idea of this thing called encephalization, which is the brain size relative to body size. And if you have a really high encephalization quotient, then that means that you've got a big brain for an animal of a given size. And a lot of primates tend to have high EQs related to other animals. These researchers now took this encephalization quotient and they changed it, and they've done the encephalization not just in relation the brain to body size, but the phylogenetic relationships around the organism in their family tree. So it illustrates a pattern within the family tree. So Chilicebus has this really small PEQ, phylogenetic encephalization quotient. It's 0.79. A lot of monkeys nowadays are somewhere around 0.86 to around 3. Humans, however, we're 13.5-ish. Woo! Look at our big brains. But this new framework, they have confirmed once and for all that this enlargement of brains occurred repeatedly and independently throughout evolution. So it's not necessary, and things decreased too. So it's not necessarily a completely linear relationship.
And I wonder if it's relevant to be looking at brain size versus blood flow. because it was one of the stories that keeps sticking with me that I was so intrigued at. And when they looked at the enlarging of the opening of the pathways of blood into the brain over time, and that they found, I don't know how linear it was really, but that was what they were sort of those researchers had been focusing on to try to determine not just the size of the brain, but actually the functionality. Like how much were we relying on it? How much blood resource were we pouring through this organ to get it to do things for us? So it's always important. While we have a 2, 3, 4, 5, that we're at the 13 quotient versus the up to 3 on a monkey, it's the blood flow goes up like 600 times over that period. So there's another element though.
There are more. Yeah. So the big point is how many factors do you involve in your analysis to really see what happened? I mean, I think the big thing here is just the real data behind it. As much as people wanted to say old monkeys had little tiny brains, new monkeys, bigger brains, humans, giant brain. It's simple. It just progressed. Now it's like, no, it didn't. And it's not even necessarily related. It has had to do with other external factors. Could be blood flow, could have been ecological niche factors that we're not even considering. Something they brought up in here as well is there's an evolutionary trade-off that has been considered kind of as fact for a very long time, that animals that see really well have visual acuteness, that they have weaker senses of smell. And there's always been thought of this trade-off. You have a big olfactory bulb, you have little eyes or a little visual system. Big visual system, little olfactory bulb. And in this Chilicebus, they discovered that was not a trade-off. They did not have this kind of trade-off. And so there's evidence now that maybe this coupling of vision and olfaction didn't necessarily happen as tightly as we have thought throughout history. So there are a lot of little things coming out of this little tiny old 20 million year old fossil remnant.
You know what I want to see now? This is intriguing me for a totally different reason, but I want to see different sized monkey brains that have similar blood flow. And see if you can see any sort of a difference. And then you need the ratio. You need the big blood flow with the smaller brain and the whole. You need kind of an array of different. But I want to see which one seems to impact an organism's life, or intelligence, based on that. That'd be interesting.
Yeah. Well, moving on from the past, let's come to the present. Humans do these awesome things. We've got the language ability. Our brain allows us to do that. Researchers at UC Berkeley, a couple of years ago, we reported on this too, at the Gallant Lab. They have been mapping where words are represented in the brain. And it turns out that we think we just have a language center, right? This language center that's what manages language, allows you to understand things or allows you to speak. Well, it turns out language is represented all over your entire brain. It's just all over the place. And you'll have the word for top represented in one part of the brain, around other words that have to do with blouse, shirt, things related to a shirt kind of top. And then top will be in another part of your brain around things like cover or roof or lid and these other meanings. So these words of similar meaning tend to map together, but in various locations all over the brain. And these locations also are kind of similar between individuals. So where these words show up in my brain is actually kind of similar to where it shows up in your brain. And in addition to that, they've moved forward now, and this research is fun because what they had people do previously was just listen to a story, and they used the storytelling program, The Moth, which is a longtime show for storytelling. You can get it as a podcast, easily downloadable, amazing stories. And people come in, sure, I'm volunteering for this study. I get to hang out and listen to the moth. Awesome stories. Sounds great. Meanwhile, fMRI, and here's where these words are showing up as blood flow activation in various areas of the brain based on time. Now, they had people read a transcript of the stories, and they mapped the brain, and they had them listen to a story, the same story from the moth, and they mapped that to the brain. Where do you think, or do you think the words that were heard and the words that were read were mapped to similar or different locations in the brain?
So this has been the thing when it comes to having a technology that can read people's thoughts, or humans being able to share thoughts through direct brain-to-brain connection. I've never believed that where I store the word blouse has any relationship to where you store the word blouse. I think they're in completely different places. Now, but what you're posing is auditory word association versus read word association, and if they're stored differently. And I would think, yes, I would think they are stored in different places.
I would guess it would depend what kind of learner you are.
Oh, very good.
Because I know that I learn way better from reading than from listening, even though I love to listen. But I feel like I would not absorb the meaning of words as quickly or as readily if I was listening versus if I was reading.
So what if it's a mix? Like you have some words that you totally comprehend immediately in a set part of the brain when you read them. And when you hear them, it's more vague. It's not the same quite. It has different connotations when you hear the word than when you read it. Huh.
Yeah.
That would be interesting too. But I don't know. Now we get
I would guess mostly different.
And this is also the thing I love about the show is that we're speculating. Kiki's got the answer.
Yeah, she has the answer.
I have
right
the answer. And surprise, surprise, the same locations.
Wow. Nice.
The words that were read and the words that were heard mapped to the same locations in the brain. All over the brain. And even though there are separate auditory systems and separate visual reading systems, there is something about language that uses all of the brain, and it's similar. We're accessing the same information when we're listening to something and when we're reading it.
So it was the same within the same individual. How much similarity was there between different individuals?
That I don't know for sure. I would need to look into that a little bit.
Because I still think where I put blouse is not where you put blouse. But it turns out whether I read blouse or hear blouse, it's in the same place in my brain. I just feel like it might be in a different place than you put it.
So my other question is, based on the way English works, we have words, right? But other languages focus on phrases.
Oh.
So there's a question there that I have about, if they're mapping as they listen to or read a transcript from the moth, are they mapping every single word, or are they mapping are they mapping?
Mopping.
Are they mopping up sections?
It's a word that Blair knows how to read.
You see, I went to the part of my brain about mops instead of mapping. No, are they mapping sections, or is it really just word by word?
They were, so what they used fMRI to record brain activity in these experiments while participants listened or read, and then created voxel-wise encoding models to characterize semantic selectivity in each voxel and in each individual participant. We find that semantic tuning during listening and reading are highly correlated in the most semantically selective regions of cortex.
So it's just during the process, just as kind of a running clock. Because I do think there's
that are more tonal.
It's meaning, though. This is the brain eliciting meaning from signals, whether it's a visual signal or a heard signal. These are signals that correlate to a particular meaning. So if it's an idiomatic speech or if it's tonal, or if, it's Germanic, whatever, I don't think it would necessarily matter so much. I mean, they've only done this in English from using the moth at UC Berkeley. This is not something that they have done elsewhere, but or with other languages. But I would think that since it's representation of the meaning of words or of phrases, that would be that is the key similarity.
It is, but there's something exciting about that too when you mentioned the tonal languages. Because if it's the same word, but tonally how it's expressed changes the meaning. Are those disparate? Now, are those, because it's just a tonal change, but do they get clustered? Yeah. That would be real. That's a very interesting question. Yeah, that is very interesting. But the words themselves, they have different accents, don't they, to indicate the tonality when they're written? Yeah, and then you also have the words that have multiple meanings even in English. And I wonder how, if
top,
they get...
for example.
Top. Mm-hmm.
If I was thinking like bat, we can be talking about something that flies or something you hit a ball with. Do they does baseball and bats get somehow accidentally
put
together in the brain because of this, or do they?
Right.
Or no, this is over in the animal kingdom. This is where we name things over here. This is a tool. This isn't with
both.
Like how,
you have bat represented with other animals that fly, bats, birds, or if your brain has put it into like rodent type things, then maybe you're categorizing it with rodents. I don't know, even though they're not. But then
like if visual morphology similarities get stuck together, it's like, how does the brain categorize? How well have we done this?
Right. These are very interesting questions. I don't know. I think the exciting part of this, though, is being able to figure out how does the brain represent language? How does it interpret language? And I think eventually this is going to influence how artificial intelligence learns to use language as well. So the chatbots are just going to get smarter based on this. I
think this is a great kind of window into why humans are so obsessed with categorizing things. Because this is how our brains work. This is why we're so focused on what group does this animal belong to, and what do I know you from, and all of these kind of categorized base. What kind of human are you? We get really focused on categories because this is how our brain works, is by putting things in neat little boxes with like things.
Yeah. A question in the chat room from Schnago wondering if words are stored in different languages for polyglots, where they would be stored. And that, I think, would be a fascinating addition to this. No idea.
I mean, like if you knew two languages, do you try to stick all the words next to the ones what they mean?
Yeah.
Would they all be associated by meaning? Yeah.
I know that when I was learning Hebrew, I had already taken French in high school and college, right? And so when I didn't know a word in Hebrew and I was speaking, I would often insert the French word in, which
because it was still in a foreign language.
Yes. And so I feel like that kind of tracks with that, right? Is that your brain is trying to complete the sentence and you're sticking the like word into that place.
Yeah, that makes sense.
Yeah, I think it would make sense.
Moving on from this study, another study of brain mapping and figuring out how things are organized. This one was published this week. I'm trying to scroll to the top of the page in Nature Communications. And the researchers here were looking at not reading and listening, but reading versus math ability. Now, unlike listening and reading, which are apparently all in the same places in the brain when it comes to figuring out meaning, reading and math have completely different areas of the brain where activation takes place. So while you're reading, certain areas of the brain are taking place. If you're computing an answer, different areas of the brain are working. There are connectors, tracts of white matter. These are the axons, the connecting cables between different parts of the brain that allow the networks of cells in the brain to communicate, so that you can have, say, that semantic meaning of a phrase that evokes a memory in you, or that you're able to understand, hey, this number isn't an S, it's a 5. I know this is a 5, and now I can add these numbers together. So there are these axons that connect the visual and the computational and cognitive areas of the brain to each other. Even though the reading and the math parts of the brain are separate, the information travels down these long axons. They're called fascicles. And they are just bundles of axons, this white matter. And they all get bundled together. So the math and the reading, those signals are traveling through your brain all together in the same paths. Now, this study did some really interesting fMRI work to dig into what was even finer resolution inside these tracts of axons and how information was flowing within them. And they found out that these, even though they're bundles of neurons, of these wires between different areas of the brain, the bundles contain little tiny wires that have different purposes. And math and reading stick to their own wires. They stay in their own lanes. So even though they're in the same bundles. If I have a math
problem it's like a word problem in math,
yeah,
it's trying to go down two different lanes at the same time.
Well, the reading part of it is going to go down the reading lane, and the math part of it is going to drive down the math lane.
At what speed? You got to tell me what speed to go. Otherwise, I can't solve it.
Well, if
they left Chicago at 4 p.m. Right, driving at 50 miles an hour, and another left Boston at 3.
Should I be in the math lane yet? 'Cause I don't
95.
feel like I switched lanes. I'm still listening to the story. Okay, where are they going?
So maybe this is why word problems are so hard, especially for younger kids in school, is that they're trying to figure out how to get these two lanes to work together.
Right, the lanes need to work together, but they are separate areas of the brain. Yeah, the information in the brain, this information is kept separate, sticks to its own lane.
I would also argue that this is why cross-cutting curriculum is so important in schools, is because you have to be working out these different areas of the brain simultaneously in order to get all of your brain working. I think that that's something that schools are doing better at, and school districts are doing better at, but it's still something that has to be worked on. Is not, okay, this is math time, and okay, this is English time. If you can get the whole brain lit up at once, that would be great.
It would be great. Let's light up those brains, everybody. I hope that's what happens when people listen to TWIS. I'm done with my brainy stories. Justin, tell me a story.
Oh, my goodness. I don't know if I got one. Oh, wait. Oh, I do. Whew.
Hope you've got more than one. We got some time.
Toxoplasma gondii, the pernicious parasite that needs very little introduction other than it blinds babies, causes behavioral changes in humans and in mice, and only completes its full life cycle in cats. Which begs the question, why cats? Not just why have cats, because there's obviously no logical reason for anybody to have a date with something that if it was just a little bit bigger would try to eat you. But why do they only complete their life cycle in cats? So new research published in the open access journal PLOS Biology answers the question, solves the mystery. The sexual phase of the parasite's life cycle requires linoleic acid, a nutrient lipid found at uniquely high levels in felines because cats lack an enzyme for breaking it down. That's it. Cats don't have the right enzyme, so it builds up, and it's exactly what this parasite wants and likes. This is a finding from Bruno Martorelli de Genova and Laura Knoll, University of Wisconsin-Madison colleagues. And they think this might lead to a treatment to reduce the spread of the parasite from cats to humans, as well as into other places. So it can live in any mammal. The Toxoplasma can live in absolutely any mammal, but for the gametes, the little sexual cells, it's cats only because It needs this nutrient, and the cat lacks the enzyme that apparently all other mammals have.
But why don't cats have it?
I don't know why cats don't have it. They're deficient, obviously. They're mutant creatures.
Now this is just getting at your dislike of cats, but
it's such a weird, specific thing.
So evil and so cute at the same time.
Do other cats also lack this? Do tigers? This is what I'm very curious.
So it says felines. So I don't know how broad they mean to paint that. I don't know. It's largely because of the human interaction around. If people did keep lions and tigers, jaguars as pets, people would live long.
I spent time in tiger den boxes.
I'm just saying, if they were a little bit bigger, we would obviously recognize the problem of keeping them as pets. I'm saying
I could have toxoplasmosis if tigers had it, because I spent time in what is basically a giant tiger litter box.
Yeah. Oh, yeah. No, absolutely.
That sounds like fun.
It was not. I almost passed out. It's it's so much ammonia.
Oh my goodness.
Yeah.
Well, cats do have a different digestive system, and they are obligate carnivores. And there's one nutrient that they have to get because they have to eat meat so that they can get this one particular amino acid. I don't remember what it is, but yeah, they're obligate carnivores because of the amino acids in the diet. And it may have something to do with this buildup of linoleic acid, who knows? But I'm just thinking it's how interesting it is if we could give cats in their food or in their water something that would break it down. Ta-da!
As long as it doesn't kill them.
Solved, right?
I mean, this is the thing. We don't know why. I mean, there could be, as Blair says, why don't they have it? There might be a thing that's like the cats need. We don't really know that. But what they did do is they knocked out the enzyme that breaks it down in mice and introduced toxo to these mice, and they got sexual reproduction of the Toxoplasma gondii in mice,
which is unusual. Which is it doesn't normally happen. So they sort of circle. That's how they the proof of their experiment. So this is also this I didn't realize Toxoplasma is the leading cause of foodborne illness in the U.S. I did not know that. That's according to the CDC. What?
The most common route of infection in humans is by consumption of raw or undercooked meat. Cat litter is a big source of this. Of course, this is why they tell pregnant women to avoid, because it causes blindness and other problems in children. Mm-hmm. This is interesting. Cats usually excrete 2 to 20 million oocytes per day in their feces.
Yep, it's a lot.
And that shedding goes on for 5 to 10 days post-infection. In ambient air and temperature, oocytes undergo a sporulation process to mature and become the T. gondii, in which become infectious T. gondii, in which they are stable then for 18 months in unfavorable environmental conditions. And they're resistant to many chemical disinfectants. So that's partly why the spread can be so infectious and if you notice, there are a lot of cats within Society of Humans for some reason.
Because they're soft and cuddly.
Yes.
They're so cute. Yes. Yeah. Well, we can maybe help. Well, the scientists are going to figure out this toxo thing. They're working on it.
This would be a big step, and I think what'll happen is that it will lead to less cats as pets.
So I don't think so.
I have an ulterior motive. I have one last thing. I have an ulterior motive for why I don't want them to fix this. And it is because this is a great reason for people not to have outdoor cats, because that fixes this problem. If they're not outdoor cats, they're not going to give you toxo. And when you have outdoor cats, they kill endangered species. So there's actually a great benefit to not fixing this problem, so more people will keep their cats inside.
Blair, that's like you're talking about like a dozen people nationwide.
No.
that are
No.
because of Toxo keeping their
Well,
cats
at least
indoors.
for example, in Australia, there's actually rules now where you can't have outdoor cats. So there's actually an opportunity to, expand that. And anyway, it's a good reason to keep your cats inside, and I would like to keep it.
How about just keep your cats inside so they don't kill birds and other endangered species? Hey, there we go.
Unfortunately,
Be a
it's not as
responsible cat owner.
motivating.
On to the next.
Next story.
Next story. High in the Himalayan mountains, there is a lake. Rupkund Lake, also known as Mystery Lake. It's a lovely lake. Actually, it's a frozen lake almost all year long, covered in ice and snow, surrounded by frozen rocky terrain in a very uninhabitable area.
Is this where we start singing that Led Zeppelin song?
Almost. And just as the ice and the snow melt away in the summer sun, it gets a new name. Instead of Mystery Lake or Rupkund, people start referring to it as Skeleton Lake. Because the lake bottom is covered in thousands and thousands of bones. And they happen to be human bones. Large-scale study conducted by an international team of scientists has revealed that the mysterious skeletons of Rupkund Lake belong to a wildly genetically distinct group of people that died at very different times. So there's a lot of stories about who these people were. It was the king and the queen and their entourage. It was this army that was passing this gap, and they got stuck in the lake. All of these stories about this being one group that had an ill-fated end. But it turns out the real story, based on what they've been able to find from these bones, is very different. This is published in Nature Communications. There's an international team of 28 researchers from institutions in India and the United States and Europe. So this is situated at over 5 kilometers above sea level, which, for those not familiar with kilometers, 5 kilometers is about, what is it? 500 meters or something like this? 5,000 meters?
5,000.
It's like a lot. It's 5,000 meters. It's about 5,000 meters above sea level. Current publication that this is a decade-long study looking at DNA and doing some deeper dives into some of the tabloids. They discovered that these people, or at least from 3 genetically distinct groups. So they did mitochondrial DNA of 72 skeletons, and a lot of those haplogroups were present in Indian populations. Some of them were from West Eurasia, and there was even one from Southeast Asia. So they then went and did whole genome sequencing of 38 of the individuals, and they discovered that 23 individuals, present day India, they could be from different regions, the populations anywhere within India. India is perfectly diverse as it is. The second largest group was 14 individuals that were most closely related to people who live in present day Greece. And another individual, again, Southeast Asia. So this little, it's not a big lake, but it's claimed hundreds of victims, and it wasn't in one event. They found that 2 of the major groups were deposited approximately 1,000 years apart. So the mystery of this, it's a very hard to get to, very rural place. There's 2 possibilities. One is people get attracted to this lake, and something about the weather can change quickly, or avalanche, and they get sucked down into this lake and they die, or they walk across and they fall through because it's hidden under the snow. Maybe they're just falling through ice and mass. The other possibility is that it's just a great place to preserve people, and people are constantly falling into frozen lakes all over the world. It's just this one.
Oh, I thought you were trying to say it was a burial ground. Like,
no,
grandma.
This one isn't that deep. So like when the snow melt happens and it kind of rushes away, like you can see everything. But in larger frozen lakes, people fall through all of it. There's like tens of thousands, and any like high altitude lake, bodies at the bottom. Just nobody's noticed it yet.
So there's, in California at least, and I guess Nevada probably too, there's a rumor that Lake Tahoe is absolutely covered in the bottom with dead bodies.
Yes, I've heard this.
Yes. But, potentially rather unfounded. But if that second kind of hypothesis could be true, perhaps Lake Tahoe is just carpeted with it.
Okay, so this is the story that I've never gotten any form of confirmation on. But there was a point in time in which the bottom of Lake Tahoe had not been discovered in. And apparently Jacques Cousteau and company went to go do some, underwater submergible videoing. And what they apparently found was, well-preserved Native Americans and a bunch of guys in nice suits from like the 50s in the cement galoshes, like standing.
Right. Yeah, absolutely.
And so they couldn't use their footage and then had to leave. I think it's a nonsense story, but it sounds good.
It sure does.
And it would kind of fit. Like that's, it's cold enough and deep enough. And why not?
It's casino town,
It's a casino town.
Yeah, absolutely. Oh my gosh. Kiki, it's time.
Is it time
Yes.
for Blair's Animal Corner?
It sure is.
With Blair?
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh my goodness. I have Dr. Justin's daughter, Dr. Poo Pills.
What? They
For
finally arrived?
koalas.
Oh, yeah.
Yes.
I forgot I did that side project.
Yes, it's a very
What?
fun side project, saving koalas. So as you know, koalas have all sorts of issues right now. Most of them have chlamydia, and they're dying of that. They're dying from habitat loss. One of the big issues with habitat loss is that there are a bunch of different kinds of eucalyptus out there, but koalas in specific areas are used to eating very specific species of eucalyptus. And so this is a study looking at the microbiome of koalas and how that impacts which species of eucalyptus or eucalypts they can eat. So they analyzed and altered microbes in koalas' guts. And the way that they did that is they actually caught wild koalas that ate only one kind of eucalyptus, kept them in temporary captivity, and then collected poo from radio-colored wild koalas that ate a completely different type of eucalyptus. They concentrated the microorganisms, packaged them into acid-resistant capsules, and gave them to the koalas, gave them some poo pills, some literal poo pills. They then found that fecal inoculations did in fact change the koalas' microbiomes. Shock and surprise. But this allowed them to eat that second kind of eucalyptus. So this could affect their ecology in a bunch of ways: nutrition, habitat selection, resource use. This is a proof of concept for the use of encapsulated fecal material to successfully introduce and establish new microbes into koala guts. So this can have impact a bunch of ways. One being that if a group of koalas are pushed into a new habitat, they could potentially be dosed with these capsules to allow them to eat the eucalyptus where they currently live. If they are taken in and treated for chlamydia and released back out into the wild, they could release them into a place that is safe for them and treat them when they're being treated for chlamydia, also for their microbiome, so that they have kind of a head start, that they have a wider ability of what to eat when they're released. Or they could just dose koalas all over Australia so that they're a little less picky. Because in any one area, there can be I think it's up to 100 species of eucalyptus. I don't remember exactly. There are so many species of eucalyptus. At the San Francisco Zoo, for example, they'll give them 17 different species of eucalyptus at a time, and that will rotate around. And sometimes they'll eat one for a little while, and then they'll move over to another one, and then they'll move over to another one. If you think about captive management, this could also help if koalas are moving from one facility to another where they have different availability. In San Francisco, we are both blessed and cursed with the fact that there is invasive eucalyptus everywhere, but it makes it a lot easier to take care of koalas. At other zoos, they actually have to ship in eucalyptus to feed the koalas. They might have a different species.
So I have a question, 2 questions,
Yes.
several questions. So first of all, I'm taking the koalas. Then they took to the new diets as soon as they had these microbes that could facilitate eating them, or would it be they would try eating the other stuff and go, it's not working?
No, they are very picky. They know exactly what they should eat and what they should not eat when they're in the wild. But when they gave them, again, this is in captivity, they basically offered them this other kind of eucalyptus.
So then
it was the only option they had. Pretty much.
The follow-up question is, could this be of benefit to pandas to diversify their diet?
So this is within eucalyptus. So it is it's still a very small range. It's very similar type of plant. Bamboo is bamboo. If you're going to try to get them to eat something else besides bamboo.
But they go in search of different types of bamboo, like seasonally or something, or whether or not they're pregnant.
But in terms of microbiomes, the giant koala and the giant panda and the red panda share the same microbes, even though they live in very different areas and eat very different bamboos. So from that, I would assume that their microbiome actually is not really influencing that choice.
Well, they have a bunch in common, but it's not identical. So then my last question is, I know you don't like pandas because they're too picky of an eater and they're dying. And do you have this similar resentment towards koalas?
I sure don't, because they're still pretty good at doing what it takes to be a species that survives. They're actually, they can be very fast when they need to. They are very good at protecting their territory.
They're trying harder.
They can reproduce quite easily. They have a pouch, which helps them to raise their babies to a good size and stature. Yeah, they're pretty good at being animals, I would say.
Unlike the panda.
Right. Yeah. Anyway.
Yeah, I think this is all really interesting, especially because there is a very important role of this kind of fecal transfer for baby koalas. So baby koalas rely on the poo of their parents, of their mum, to be able to get the microbes that allow them to digest the food that they're going to eat as adults. And so they get like tied into these very particular trees and food types. And and then let's just give you some new poo so you can eat new trees.
Yeah, I would say that does make this pretty specific to koalas. So I do like that you mentioned that as well, is that, when they're babies, they're getting the seed bacteria from their mom. So they are a perfect poster child to start this with. There's still an opportunity potentially to help expand this. And it could help with captive management of various endangered species. It could help with releasing endangered species into new spaces. If there's not if you want to release a particular subspecies of giraffe that's used to eating a very specific kind of acacia, and you want to release them into a place that is safe for them but doesn't have that specific kind of acacia, maybe you can seed their microbiome. There's lots of opportunities there.
Yeah. So many. From poo, where do we go from here?
Oh my goodness, I'm so glad you asked. We're going to go jumping and falling right onto our face.
What?
That's how locusts do it anyway. So locusts are an example of an animal they're really good at jumping. They're really, really, really fast. They're really good at jumping to escape predators. So researchers, I absolutely love this, wanted to look at exactly the mechanics of how they are so good at jumping. And so they took some high-speed video and they found out that locusts can precisely control the movement of their hind legs. But when they land, it does not look quite so graceful. And I am hoping that Kiki can show us some video here. Yes. They plop right on their face.
Yeah, it's a little front heavy there.
So those of you that are very loyal audio listeners to the podcast, I would encourage you to go to TWIS.org/YouTube and look at this section of our show in video, because I've watched this on a loop for so many minutes. It's fascinating to me. They just plop right on their face. But why? So they took locusts in this study. They did this high-speed video, and then they did high-speed video of a dead locust and of a cold locust. Now, quote, cold locusts have only very limited control of their limb movements, very similar to a drunk person. So they wanted to see kind of how did they fall? Did they fall on their head? How good were they at getting back up and ready to jump again in these different states?
I bet the dead locust came in last.
Yeah, so I'm hoping he could show that video too. It is so great. So the insects can actually, these locusts can rotate their body in midair. No matter how they were held before they were dropped in the lab, they always fell head first towards the ground and landed in such a way that they were pretty much prepped and ready to jump again almost immediately, the way that they crashed down after they hit their head first onto the ground. This creates what they consider predictable body posture. So this just makes it easier for them to reorient to jump again. If you know how you'll fall, you can prepare how to respond after initial impact. So with the cold locusts, they did a lot of uncontrolled flailing, kind of trying to right themselves. They still were able to get back up to a point where they could jump again pretty easily. The dead locusts, they just dropped aimlessly and just they even bounced.
They were dead. They were dead. Fell.
But they all hit head first, which also means that is the energetically easiest way to fall.
It's the heaviest part of their body too, and so aerodynamically they would orient that way, right?
Yeah, so this is the passive role of the insect body when falling. So if they can know that and they can adjust exactly how they fall on their head, they're not expanding energy trying to move around as they fall. So a couple things about this. One is they're hoping that they can use this information to create better response in probes and drones and robots and how they respond to falling or landing or crashing. If you think even about the landers that we land on other planets, if you, instead of focusing, kind of the idea here is instead of focusing on a soft landing, focusing on maybe a harder landing, but predicting how that landing will go, padding that area because that's their situation, right? They have a exoskeleton that's really robust there on their head. Padding that area and being able to predict how that crash will happen and responding to impact instead of trying to engineer a soft, perfect landing is a different way of looking at it. And that's how nature does it in this case.
It's okay if I crash. I'll be fine.
Yeah, it's a controlled crash, right? That's exactly what this is. To say, instead of saying, I'm going to try to slow down and just barely boop, hit the ground, I'm going to drop this probe 1,000 times and know statistically exactly how it's going to smash when it lands. And then I can, beef up that area and put out, I don't know, like almost like kickstands to respond when it falls backwards from where it lands on.
Right.
So there's a new idea here and how to engineer that.
Yeah. I love it. I'm thinking of that line from Toy Story when who's the space guy, the space toy?
Buzz Lightyear, and he's like, I'm falling with style. Yes, exactly.
Yeah.
Just
but
the
in
best
this case,
style.
In this case with the locusts, it's falling without much style.
I think it's great style. They're like, I don't care about my head. It's just a ball of neurons. It's not even really a brain.
That wouldn't work for people quite the same way.
No, not without a really good helmet and neck support.
Yeah. Well, we're going to keep doing this show with style. Yeah. That's right. Is it time for us to take a break?
It sure is.
All right, well, let's take a quick break. We will be back in just a few moments with more This Week in Science. I've got more brains, and Justin still has Mongolia. We'll be back with more TWIS.
explain things you've heard with more than intuition.
Thank you for listening to TWIS. If you would like to see and hear more of TWIS, well, you can go to twis.org and subscribe to our newsletter. That's right. If you go to twis.org, there is a pop-up screen which should get you the ability to subscribe to our newsletter with your email address and your name. And once a month, we will send you our newsletter that's full of extras from the TWIS team, little thoughts and noodlings and ideas. And it's just a fun little addition that we're doing. Beyond our newsletter, at our website, you can find ways to subscribe. The big orange subscribe button will get you to YouTube, iTunes, or Google Play. We are available other places, but those are the big three. So if that's where you want to subscribe, it's an easy way to find those links. Go to twis.org, click subscribe, and choose your platform. We are also on Facebook as well. You can also head over to our Zazzle store if you're interested in some of our merchandise. We have wonderful merchandise. We have shirts and tote bags and hats and calendars and all sorts of wonderful things with our logo and artwork from the wonderful Blair. She's working on a new calendar now, everybody. Isn't that exciting? A calendar for 2020 is coming. But if you want some of that art, you can have it on a keychain or on a mug or on a pillow. That's right. It is. It could be yours. Zazzle.com. The link is at twis.org. Back at twis.org. Also, This Week in Science is an independently run podcast. We're not part of a podcast network. We are not owned by anybody else. It's just us, and we do this show on a weekly basis, and if you like what we do, we would really appreciate your support. If you are interested in supporting us, an easy way to do that is to click the donate button. It takes you to a PayPal interface. If PayPal is a way that you like to support podcasts, that could work. But Patreon is a wonderful community where you can choose the level of your support, click the red Become a Patron button, and everywhere from $3.14, cute, high support, all the way up to $50 a month, you can choose your level of support. You're charged once per month, and that support goes directly to us being able to do the show, buy new equipment, be able to do the show better. And this month, we're actually looking at an increase in some of our finances. Blair's computer's pretty much on its last legs. It keeps dying, and we need to get her a new computer. We're going to go in halvesies with her, but we need your help to be able to do that so that she has a strong enough machine to be able to handle the streaming and the video and possibly some video editing and audio editing and that kind of stuff. Also her art. There's a lot of things there that computer would contribute to with respect to TWIS, and we would love your help being able to make that happen. Also, we're using StreamYard now to broadcast the show and to be able to get rid of the StreamYard branding and be able to add some other bells and whistles. That would be pretty awesome. We're going to have to subscribe to the amount of $20 a month. There have been some wonderful patrons who have already increased their levels of support. Thank you so much for doing that. But to be able to afford that additional $20 a month, it's not too much more, but it is that little tiny expense that does make a little bit of difference. If you can help out, it would be much appreciated. You know, we do this show week in and week out. We do it for you. We do it for ourselves too, because it's pretty fun to talk about the science, and it gives me a little dopamine boost, and I get really happy on Wednesday nights when we do the show. But it really also, I love connecting with you. I love seeing people in the chat room. I love getting emails in messages throughout the week. I love seeing you on Twitter, Facebook, wherever it is that we connect. And I just really appreciate the fact that you are a part of TWIS. Couldn't do this without you. Thank you so much for your support.
We can explain things you've heard with more than intuition.
And we're back with more This Week in Science.
Oh, yeah, we are back, and we have more science for you. Oh, my goodness. But first, it's time for This Week in What Has Science Done For Me Lately? This week, we have a letter from, let's see if I can get my my window to tell me the name. I can't read it.
Oh, it's Eric from Alaska.
Eric from Alaska. Thank you very much. Yes, Eric from Alaska. Eric and AK in the chat room. He writes in and says, "Hi, Kiki, Blair, and Justin. I have written 2 What Has Science Done for Me Lately stories. Previously, one was about an illness and the other was about a natural disaster. This one is not like them. This will be a happy one. The science and technology of telecommunications and shipping have allowed me to more fully enjoy my hobbies. I am a collector of stamps and of Boy Scout books. The sales and auction sites on the internet have allowed me to purchase items I would never have found any other way. I have found information that would have required a trip to an archive in a faraway country just a few years ago. I have also made hobby friends all over the world. I often repeat the old joke that there is a tiny difference between a hobby and a mental illness, but my hobbies have helped me in many ways over the years. When I am working on my hobbies, my mind is calm, I am engaged, and I am enjoying myself."
Nice.
So science has given me tools to make my enjoyment of hobbies better, and for that, I am grateful. Thanks, Eric. Yeah, science, making our hobbies more enjoyable.
I was just thinking, Indiana Jones's job would have been a lot easier after the internet. He spent a lot of time going to far-off libraries and stuff.
Yeah. That's right. There is a certain amount of, and also the interlibrary loan. Those are always good, but they take a while. But having those documents available digitally through libraries, amazing. The internet connects us. It's amazing. If you want to talk about the connectedness of the world because of science, or you want to send us a note or a sonnet or a little story, something telling us what science has done for you lately, send me an email, Kirsten@thisweekinscience.com, or send a message on Facebook. You can find us on Facebook, This Week in Science. All right, time for more science. Justin, what's your next story?
Underwater archaeology via the Maritime Archaeological Trust. They have just discovered an 8,000-year-old structure next to what is believed to be the oldest boat building site in the world on the Isle of Wight. This is Director of Maritime Archaeological Trust, Gary Momber. This new discovery is particularly important as the wooden platform is part of a site that doubles the amount of worked wood found in the UK from a period that lasted 5,500 years. The site lies east of Yarmouth. Yarmouth, if you are from the UK, you might not even know where that is. And the new platform is the most intact wooden Middle Stone Age structure ever found. Yes, this is Stone Age. Usually when we think of the Stone Age, we think, the modern material of the day being the same as it ever was. Everything's just made out of stone. That's how we make things. We build them out of stone. But this is, part of the Wood Age right here. So the site is 11 meters below sea level. There was during the period when there was human activity on site, it was a dry land, lush vegetation, and this is before the North Sea was fully formed, and the isle was actually still connected to mainland Europe. So this is sea levels were quite different. 11 meters, that's 33 feet. That's a big difference when it comes to sea level in just that 8,000 years. Site was first discovered in 2005. Contents and contains an arrangement of trimmed timbers that could have been platforms, walkways, or collapsed structures. Kind of difficult to interpret until the Maritime Archaeological Trust did some underwater archaeology. They went down, they took 3D models of landscape, and they began the excavation. Gary from previously, Momber continues, the site contains a wealth of evidence for technological skills that were not thought to have been developed for their couple thousand years. There were advanced woodworking skills on display. The site shows the value of marine archaeology for understanding the development of civilization, which is something I've said many times on the show before. That's where we have to discover the history of civilizations is underwater. Yet, being underwater, and this is something I hadn't thought about, there are no regulations that can protect the site. Therefore, it is down to our charity, with the help of our donors, to save it before it is lost forever. So it hasn't been fully removed yet. It's one of those. So again, this is one of those things. Again, I've talked about it a number of times on the show, but humans tend to live in coastal zones, right near the sea. We are the son of the son of the son of a fisherman, and the daughter of the daughter of the daughter of a fisherwoman as well, and the son of a daughter. Well, and the sea level rise for a very kind of important technological transformative period of our history is now underwater. It's going to be out to sea. I'm glad that they found this site. It's an amazing discovery to find woodworking skills that predate by thousands of years, where we believed them to have been. And it's also one of those things, like if boats are also around earlier than we believed them to have been, it helps explain some of these strange leaps where people showed up. Yeah. And one of the things that's always tough about this versus any other technology that we possess is things made out of wood decay. So if we did have boats even many thousands of years before this, the likelihood of finding one preserved is very unlikely.
So we've had a space archaeologist on the show. I think we have to have a marine archaeologist. I don't think we've had that, at least in a while.
All right. We will put that into the little pot of the magic pot of names, ideas to pull out and create it. We will try and make that happen. I like it.
If you're a marine archaeologist listening, hit us up on Twitter.
Yeah, let us know. Join us on the show. Be a little reckless.
Yeah.
Reckless? Why would somebody be reckless, Kiki?
Oh, reckless like a teenager with poor impulse control.
I feel like there's some science related to this.
There is some science coming at you right now. A paper published this week in Trends in Neurosciences looked at not recklessness necessarily, but impulsivity control in macaque monkeys and in humans. They compared adolescents of both species to adults of both species on a task that was basically like, here's this thing, don't look at it. And adults were much more able to not look at the thing that they weren't supposed to look at, whereas the teenagers or adolescents were like, I've got to look at it. And they were worse at being able to control or stop themselves from acting on the impulse. Now, anybody who knows teenagers just kind of go, well, this is just part of it. We know the prefrontal cortex is the last part of the brain to mature. This is the area involved in impulse control and decision making. And the researchers conclude from their study that since they found the same trend in macaque monkeys, which are distantly related to people, that this is an evolutionarily conserved trait that must have some kind of importance in our evolutionary history.
So it's being described as impulse control. I immediately thought that's just curiosity and having an imagination that can create the idea that that's something interesting enough to forget what somebody just told me to do. Whereas an adult is like, there's no secrets. I don't have an imagination. I'm not even curious. They told me to stare at a blank wall. I'm just going to do that. I don't feel like that's a good impulse quotient.
To me, it sounds more like risk-taking behavior that I think it's part of normal human development. They talk about it as a benchmark in the cognitive development of children, is learning how to take risks. Taking risks is an important part of developing your brain, and then evaluating risk and adjusting whether or not you're going to take a risk based on how high the risk is. This is like a really important thing to learn as a human. It makes perfect sense to me that actually not just primates, a lot of animals, potentially all animals, would need to learn this when they are younger, when there's somebody looking out for them, or they have more safety nets to help them learn what is a risk worth taking. If you even just think about like the deer going out into the clearing, when is it a good time to go into the clearing? When is it worth it?
Right. And, that's exactly the perspective that these authors take on this conclusion, on their interpretation of the results, that the risk taking is valuable because it leads to more learning experiences that would not take place if individuals were completely risk averse from, a very young age. So it's learning to control the impulse of risk taking, to control your ability to do something or not do something. Plenty of adults still have issues with their impulse control, but the decision making, that risk taking, the choosing to do something or not, being able to control it, and having the learning experiences that occur as a result of that process is valuable to your survival as an adult.
Yeah, that's why like doing stupid stuff when you're a teenager is really important, actually, and getting hurt.
It is to a degree. It's like if you kill yourself, you've taken yourself out of the gene pool. Right. It's not working. It's all within limits, right?
Yes. It's you have to learn fire is hot. There's this level.
You can't just go, somebody told me fire's hot, and then that's how you live your life.
So
okay.
This puts impulse control in a strange category, though, because in a way it's putting impulse control in a thing that's an ability to override a thing that is in your best interest. Which is like, the deer.
I don't think impulses are always in your best interest.
No, but in this case, if the impulse is to learn, is to take that chance to learn something new, to see the thing,
But there's a certain point where maybe you haven't learned everything, but you've learned enough lessons that you know how to react to things. You think you know how to better respond to situations, and you don't need to just try something. You don't need to do the risky thing. Yeah. But anyway, they're thinking it's evolutionarily beneficial to be impulsive when you're young.
Makes perfect sense to me.
Yeah. Anyway, Justin, tell me another story.
Oh my gosh. Okay, so the last story I brought is now Mongolia.
All right, Justin bought Mongolia. I forgot.
I bought Mongolia. Yeah. So again, we're going to the Stone Age, right? Where the modern material is the same as it ever was. Once again, and they found in the Stone Age in Mongolia Stone Age tools. Okay, I know you're thinking that's what you would expect. Except these are 45,000-year-old stone tools and the Eurasian steppes. That's about 10,000 years earlier than archaeologists previously believed that people were in this region. So it's a big pushback. It also then creates a new point of possible encounter and contact with Denisovans. Because they were still there in that. So this is Nicholas Zvins, associate professor of anthropology, lead author of the study. He led the excavations between 2011 and 2016 at the Tolbor 16 site along the Tolbor River in northern Hengai Mountains between Siberia and Mongolia. They yielded stone artifacts. 126 of them were associated with the oldest human occupation site there. There was long, regular blades, tools that resemble those found at other sites throughout Siberia, northwest China, including a large scale dispersal of humans across this region. So there was, and they were well crafted too. It was one of the interesting things that they sort of pointed out is that was as if there was a very systematic way in the similarity of all of these tools that they were being created. So there was a large group of humans that shared a very significant technological, cultural background, and it wasn't, these weren't disparate groups throughout the region. So they found no human remains at the site. They have the tools. So there's even maybe a slight possibility that these are not from humans. Well, okay, wait. The age of the site determined by the luminescence dating on sediment, radiocarbon dating of animal bones found near these tools. Is that 10,000 years earlier than the fossil of a human skull cap from Mongolia, and roughly 15,000 years after modern humans left Africa. So apparently this was the first stop. People left Africa on one of the migrations out. Now we kind of seem to know that there's been more than one. And there has been some talk of, there was, I think it was the Nat Geo, was it the Nat Geo Genome Project that found a Mongolian man that was like the most related to the most people in the world.
Right. Yes.
So there is this sort of unique position for the Mongolian people there as well. And this is quoting voice from Zwins. Although we don't know yet where the meeting happened, it seems that the Denisovans passed along genes that would later help Homo sapiens settling down in high altitudes and to survive hypoxia on the Tibetan plateau. From this point of view, the site of Tolbor 16 is an important archaeological link connecting Siberia with northwest China on a route where Homo sapiens had multiple possibilities to meet local populations such as Denisovans. So
that's cool. So that's maybe where along the way we ran into the Denisovans, intermingled, and continued our migration around the planet. Huh.
And again, this we find a 45,000-year-old site like this because it's on the on the Mongolian plateaus, right? It's on land. This is not where most of the people were back then. They were coastal, and that coastal is now underwater. That's why we need the underwater archaeologists to join our show.
There's so much to learn along these coastlines. Oh my gosh. So much being lost, so much to learn. Learning, though, so there's this idea, the 10,000 hours of practice, and you're an expert. You're going to know how to do something. There's a study that came out back in the early 90s by a group, Erickson Kramp and Teshk Romer. I can't say the name correctly, but this was a psychological review study, which is a study that so many people have cited and used as evidence for things they're doing. This study looked at, back in the 90s, looked at violinists and this thing called deliberate practice. How much do you have to practice to be really good at something? And they found there was a direct correlation. People who didn't practice very much, they weren't good. And then if between people who were considered good violinists and expert violinists, there was a difference in this study. The more you practiced, the better you were. Now, in the study, they had a delineation between this deliberate practice and teacher-designed practice. And the research, there were a few questions around this that led some new researchers, Brooke McNamara and Megan Matra, to repeat the study, but with a few of what they think are improvements, like one, employing a real double-blind procedure so that nobody knew what group they were in for the analysis. Conducting analyses better suited to the study, they say, and testing previously unanswered questions about teacher-designed practice. That they examined the way Erickson et al. Operationalized deliberate practice, that's practice by themselves, and their theoretical but previously unmeasured definition of deliberate practice that was teacher-designed practice. And they compared them. And so they, in the end, did not replicate the finding of this seminal study from the 90s that everybody has been like, just practice. Practice is the thing.
Is a lot of hours.
Yeah. I mean, I can definitely say, as someone who spent a lot of time in bands, there are people who just, you put the instrument in their hands and they have an inherent skill.
Yes.
It's a thing. I mean, especially if you think about improvisation and stuff like that, you have an ear. Some people have an ear for it.
Yeah. And so whereas this early 90s study democratized abilities, and basically people used it to say, just the more you practice, if you practice makes perfect. The more you practice, you're going to do it. All it is practice. It doesn't have to do with anything else. This study, by making the changes that it did to the methodology, they were able to delineate that there actually is a difference between the expert, really good violinists and the good violinists, and it's not just practice. In fact, they found the very, very best violinists practiced less. Than the others.
Yeah, all the prodigies in my bands, I would ask them, God, how much do you practice? They're like, I don't know, like maybe once a week for an hour.
Yeah. And so there is...
Show up and do that.
This study did not look at genes or anything to do with inherent ability, but by reanalyzing, by reworking the study, they do have a very, not definitive, but there is a line to this idea that, yeah, there is genetic and inherent ability involved in at least that complete expert level of ability, where people are elite athletes, elite musicians. There is something to the idea that there is genetic superiority in some domains.
So to throw this a little bit into this too, if you're designing a study, and they didn't realize this study was going to be this big when they started, of course, they had no idea this many people would pay attention and be like, this is how you do it based on this one study. Everybody must practice 10,000 hours. But when you're conducting a study, remember that it might become important in the future. And people might look back on it. And so just make sure you've really got it ratcheted down, what your definitions are, what the parameters are, and that you do it there. There was some story I heard today that Mendel had a pea study.
With peas, yes.
With the peas, that the peas may have been more influenced by the gardeners who knew what he was looking for than anything else, because something didn't show up in his study, which was a crossover of genes. He got one or the other of the pea, but there was never evidence of a crossover, which when you repeat the experiment, you always see. So, yeah, people, you put this document out there, it's forever. Because if it becomes important, if anybody starts to really look at it and thinks it's important, it will get looked at. And so, yeah, just be careful when you're designing your experiment.
Well, I mean, all of the band teachers out there really have to rethink their pep talk now.
Oh, they know.
I certainly got this one plenty of times. Like, it doesn't matter if you.
Well, it does. So the bottom line is, even though there was this less practice result for the elite, the really good violinists in this study, the difference between everybody else was based on practice. So. Right. If you're just an average person, want to learn an instrument, you're not going to learn it if you don't practice.
Right.
And things take practice, and you will improve with practice. But maybe what this study is saying is that you might not end up, right? You could, there's all sorts of things.
So
practice. Practice.
I have according to 23.
I don't want to put people down. So
no, it's true. I mean, you can get to be really, really good just with practice.
With practice.
Yeah. But you still might not get first chair because somebody else has some innate talent.
I have a gene, according to 23andMe, that I share with the elite of the elite athletes. And this gene gives elite athletes about a 3% advantage over other elite athletes without this gene, which makes me 3% more athletic than a couch potato. because I haven't done the other 100% of work that the elite athletes have done who don't have this gene. There is no free ride even within the genetics. You still have to participate in the world. Yeah.
You have to participate. That's right. It's a participant's world. I have a whole bunch of really quick stories. Can I run through them? I'm just going to run through my quick stories. This one's for you, Blair. Spiders.
Spiders.
Yeah, my first story of the quick stories for the end of the show. Apparently, according to some researchers from UC Davis and UC Santa Barbara, these guys went following spiders in 2018 after the hurricane season. They were looking at a particular type of spider, Anelosimus studiosus. This is what's known as a comb-footed spider, and it's known for exhibiting aggressive or docile behavior. These are colonial spiders, and sometimes the whole colony is aggressive or the whole colony is docile. And these traits get passed down from one generation to the next. So if aggressiveness becomes part of that generation, then the next generation is going to be aggressive. They followed the hurricanes from the 2018 hurricane season and the spiders and looked to see who survived. It turned out that the more aggressive spider colonies were the spiders to survive. Nobody knows why, but that's just the way it worked out. The docile spiders didn't do well after the hurricanes. So now they have to find out exactly why. Their methodology is pretty awesome. They take an electric toothbrush and they go vibrate the web of the spiders with electric toothbrushes and then wait to see which spiders come running out. And the ones that come running out, they label aggressive.
That makes sense. Yeah. But also now there's going to be predominantly aggressive spiders in these hurricane areas.
Yeah, for a while until it goes away for whatever reason. Yeah. Yep. So aggressive spiders becoming more aggressive, colonial spiders becoming more aggressive. Just what we wanted, right?
Cool.
Yeah, this story was sent in by Artyom, and in case you hadn't heard, there have been a few tests of various Ebola treatments, and we know that Ebola has been ongoing in the Congo and other regions of Africa. This is a huge human health concern. There's a vaccine that does seem to work, but if you haven't been vaccinated, what do you do with the people who've been treated? So they've been looking at monoclonal antibodies of a couple of different treatments, and all of the trials are looking really good. So they're taking the mortality rate from Ebola down from about 75% chance of death if you get Ebola, down to about 40%. Or possibly even 20-ish, depending on how soon people come in after they start showing symptoms.
So market improvement.
Yeah. It's a big improvement. It's not a cure, and you might hear people talking about it's a cure, but it's not a total cure. But this will save a lot of lives.
I would take those odds for sure. Yeah,
and the big issue now is just getting people to come into the clinics, because that is something, and to get treated, because there's a lot of distrust in the medical institutions. Another study out of the LIGO and Virgo arrays looking for gravitational waves have detected a black hole swallowing a neutron star, and this was pretty much the trifecta that astrophysicists were looking for. We had a black hole running into a black hole, we had a neutron star running into a neutron star, and now they have detected a black hole devouring a neutron star. They're confirming all of their measurements and results, but this is going to be giving astrophysicists some really wonderful information about these massive objects in our universe. Very exciting. Yeah, and my final study has to do with the brain because it's super brainy night for me. Neuroscientists looking at an area of the brain called the hippocampus, which is my favorite because it lets me remember stuff. It lets you remember stuff too. The hippocampus is responsible for the consolidation of your memories. It puts them places, and we've known that there was this signal. It's called the sharp wave ripple in the hippocampus when information starts being stored. And they have just detected this sharp wave ripple in the hippocampus 1 to 2 seconds before a memory is actively recalled by a subject. So now they're wondering if this is part of the memory trace, if this is part of the brain accessing its distributed memories when they are being recalled. But we don't know. They don't really know. All they know is that there's this there's sharp wave ripple 1 to 2 seconds before memory recall, and that's all they know. They don't actually know that. They don't know if it's connected to anything at all, really. So, yeah.
So
anyway.
isn't that 1 to 2 seconds, though? Isn't that a while?
That's
is that a tough memory?
That is a very long time period when it comes to stuff in the brain, because there are lots of brain signals, various frequencies that constantly firing, and you know that you're looking for a particular signal that's going to happen at a particular time, and there are different waves that can be detected milliseconds before things that are very important for various brain functions, for brain activities. But
quick question, Blair, right now, where are your keys? 1,000. 2.
They're on the key hook.
Oh, well, maybe that is no longer taking for a memory. Maybe actually that would seem okay. I get it.
I'm tired.
Yeah. I love the introduction to the abstract on this science article, though, for this. In the abstract, they say, perhaps the most glaring unknown is the relationship between SWRs, sharp wave ripples, and conscious cognition. We still do not know what cognitive process, if any, is linked to the emergence of SWRs. To put it simply, we still do not know what an animal thinks about, if anything, when the hippocampus elicits a ripple.
Well, so there is a weird thing there that I think that they might be mentioning, which is the conscious self of us running around with the brain firing constantly going, hey, I want to access something specific that I'm not thinking about right now. Go find it. It's like a little search browser that you open up and be like, remind me about how this works. And now the brain's got to go like, you weren't talking about a completely different subject.
I've got to go to the librarian.
Yeah. Now you got the card catalog and the microfiche. We'll find it. Just give me just give me a second.
Give me a second.
Yeah. Interesting.
Yep. That is interesting. Blair, do you have a last story?
Yes. Speaking of finding things, a serendipitous discovery by some marine biologists from the American Museum of Natural History found a school of thousands of flashlight fish while scuba diving off of the Solomon Islands. Only around 25% of the world's fish species school, and as far as we knew, because these schools disperse at depths with critically low light, it required ambient light. But this is the first time ever bioluminescent flashes were observed to coordinate movement and facilitate schooling behavior in complete darkness. So this is a brand new discovery, could completely change the way we think animals behave in the aphotic zone, and reveals a completely new function of bioluminescence.
Ooh, coordinating activity. I'll flash over here. Yep, I'm going to catch up. Wow, it's like Fish Burning Man.
Mm-hmm.
Make sure you have your blinky lights before you go out on your bicycles.
There you go.
Burners at night.
I can only imagine.
Yes. It is almost, oops, there's my show notes. It is almost Burning Man for those burners out there. May you all enjoy your next week in the dust. We have come to the end of our show. We've done it. Come to the end, and I would love to say thank you. So, thank you. Thank you for joining us for this episode of This Week in Science. Thank you to some particular people for making this show a little bit easier to do. Thank you to Identity 4 for recording our audio for our podcast. Thank you to Gord McLeod for helping to manage the chat room, our web chat, our free node, web chat free node chat room. It's #TWIS if anyone's interested in joining that. And thank you to Fada for every week helping with social media, with show notes, and with the YouTube chat room. And thank you to our Patreon sponsors. Thank you, Paul Disney, Andrew Swanson, Richard Onimus, Ed, Andrew Groh, Stu Pollock, Philip Shane, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Craig Landon, Mark Mazzaros, Jack, Matthew Litwin, Jason Roberts, Bill K, Bob Calder, Time Jumper 319, Eric Knapp, Richard, Brian Condren, Dave Neighbor, Matt Bass, Darwin Hannon, Patrick Pecoraro, Ben Bignell, Jean Tellier, John Gridley, David Williams, Corinne Benton, Adam LaJoy, Sarah Chavis, Rodney, Tiffany Boyd, John Bertram, Mountain Sloth, Seth O'Gradney, Stefan Albaran, John Ratnaswamy, Dave Friedel, Gerald Myshak, Paul Ronovich, Sue Doster, Dave Wilkinson, Richard Porter, Noodles, Kevin Reardon, Christoph Zucknerek, Ashish Pants, Ulysses Adkins, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Suzuki, Jim DePro, Greg Riley, Sean Lamb, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert, Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, and EO. Thank you for all of your support on Patreon. And if you are interested in helping us out, helping us to meet our financial responsibilities for this show, you can find information at patreon.com/thisweekinscience, or just click the Patreon link at twis.org. And remember, this show grows when you recommend us to your friends. On next week's show, we will be back broadcasting live online again at 8 p.m. Pacific time on twis.org/live, where you can watch and join our chat room. We have a guest next week. Dr. Rochelle Tanner will be joining us to talk about nudibranchs and climate change and all sorts of oceany things. I'm very excited about that. Blair has made this connection for us. I'm so glad that she has. It's going to be a lot of fun. If you can't make it, though, don't worry. This is a podcast. You can listen to it when you find it, when we come to your phone or your device, whatever it is. We're a podcast. We're also on YouTube. You can find us, twis.org.
Thank you for enjoying the show. TWIS, if you wherever you found us this time, you can come back to this place, or you can go to any of the places that Kiki described and find us that way.
For more information on anything you've heard here today, for example, some videos of those locusts falling on their faces, show notes will be available on our website. That's at www.twis.org. And while you're there, you can also make comments and start conversations with the hosts or other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line. Otherwise, Blair, what will happen?
You'll be spam filtered into oblivion.
You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there is a story you would like us to cover or address, a suggestion for an interview, a haiku that comes due in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from this show, remember,
it's all in your head.
This Week in Science. This Week in Science.
This Week in Science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of Toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science. This Week